Good afternoon. I'm going to talk about our study on the therapeutic effects of pHIFU combined with chemo drug-loaded nanodroplets for treatment of prostate in vivo using a mice model. The ideal drug delivery system for cancer therapy should have the following features. Drug should have long circulation time. This one can be done using biomaterial coating, such as using PEG to avoid recognition by the reticular endothelial system. For example, this is for phagocytes. And also, the drug should be stable when it's circulating. And the size allows to extravasation to the tumor by EPR effect. And also, the drug should be released locally, for example, by using ultrasound. And the drug's biodistribution should be monitored. So based on all these features and the requirements, we have developed nanodroplets, drug-loaded nanodroplets. This is a schematic diagram of chemo drug-loaded nanodroplets from our study. The central core can respond to ultrasound to create microbubbles. The red shell loads chemo drug with biomaterial insoluble stuff. The outer shell can be used to avoid recognition by the immune system, such as phagocytes. So the formulation of our chemo drug-loaded nanodroplets is shown here. This is the nanodroplets we used. View under microscope. The size and the biodistribution for the study of our nanodroplets has been accepted for a poster presentation at this symposium. I'm going to present our results from our two studies. In study one, the animal were treated with pHIFU and with chemo drug-loaded nanodroplets. We used paclitaxel. And the second study, we used docetaxel loaded nanodroplets with HIFU. In study one, linkable cells implanted in the prostates of mice. Tumor-bearing mice randomly divided into 4 groups. In group 1, animals were treated with paclitaxel encapsulated nanodroplets with HIFU. Groups 2, animals were treated with HIFU alone. For group 3, the animals were treated with drug-loaded nanodroplets alone. For 4 group, the animal was used as a control. Our ultrasound parameters are 1 MHz, 25 acoustical power, 10% duty cycle, and 60 seconds for each sonication. After treatment, the tumor volume were measured weekly on MR for 4 weeks. This is our experimental setup. We use Encapsulate 2000 Incytec treatment system, and we also use a 1.5 Tesla GE MR system for our MR-guided high-intensity focused ultrasound treatment. This is the results from study 1. You can see with the HIFU alone treatment, has a therapeutic effect in tumor control. And for the group with nanodroplets alone, this group, and has a significant in tumor growth control. For the group with HIFU and nanodroplets, we can see the most effective treatment effect in tumor control. It's statistically significant with a p-value of 0.04. 0.004. In order to confirm whether these results are reproducible, we did a study 2. In this study, the study design was the same as study 1, except for using a different chemo drug. This time we use docetaxel nanodroplets. And also we added one free docetaxel injection. The study 2 and the results show that the results are consistent with our previous study 1 experiment. You can see the group animal were treated with nanodroplets plus HIFU, this group. And then the tumor growth control is significant with the p-value of 1.039. In conclusion, our animal study showed a great potential for prostate cancer therapy using chemo drug-loaded nanodroplets activated by focused ultrasound. More experimental studies are needed to confirm these initial results. The role of pHIFU in targeted drug delivery needs to be further investigated. So this study was supported by Focused Ultrasound Surgery Foundation Research Award and in part from grant DOD. And we would like to thank Incytec for excellent technical support. Thank you very much.
Any questions? So given these were animal studies, I don't know, did you see any significant adverse events in the animals that would be of concern? And overall, at a higher level, would you expect the adverse events to be better, worse, or about the same as what we would give when these drugs are given IV?
That's a good question. No, we didn't see any advance in animal study. No single animal died after treatment because this is low. This is a high intensity, but it's pulsed. The temperature is not high. It's just below like 41 and with temperature mapping. Yeah, skin is perfect. We don't have we didn't see any skin burn or something like that.
In your studies, you show that HIFU is almost like the control, like the HIFU is doing nothing to the tissue, while usually HIFU do ablate tissue and have in clinical practice in human 90% destruction of cancer, while the drug you are using in human is very low in controlling the disease. So what is the secret of the drug is giving more effect than the HIFU?
Okay, let me just go back the results. From the first study, we can see the HIFU group has some therapeutic effects. And then the second study, the results are consistent. Actually, we have a separate study on HIFU alone for tumor control in vivo. We also use atopic prostate cancer model. So this paper is already published in medical physics. If you are interested, I can give the reference. We talk about all the possible mechanism for HIFU alone group, why it's effective for the cancer.
Let
Control.
me clarify one thing, and that is you used the 25 watts total acoustic power.
Acoustic
And
power, that's
what
right.
was the duty cycle? How long was the pulse on? So was this a therapeutic
The
dose
pulse
or
is
subtherapeutic dose?
10% duty cycle.
So it's
Pulse rate
therapeutic.
is 1 hertz.
So it's really not to mean ablate the tissue.
Yeah,
Okay.
it's not ablated tissue. Perhaps there is some cavitation involved. This is still need to be investigated.